欢迎收听新化学家播客，很高兴您收听，欢迎订阅并收听 Apple Podcasts、Odyssey、Spotify 或任何其他收听平台。欢迎收听播客，在这里，好奇心与创新相遇，探索无疆界。加入我们，与世界知名的思想领袖交流，包括校长加里梅、罗伯特·兰格博士、格雷格·扎克曼、《华尔街日报》、凯尔·沃恩、福布斯30位30岁以下精英榜单等等。通过深入对话，我们将探索科学、领导力和目标的变革力量，从开创性的研究到改变生活的创新。请注意，本播客的观点仅代表我和嘉宾本人的观点。此外，这些观点不构成医疗或专业建议，请咨询相关专业人士。我们不承担由此造成的任何损失。
欢迎收听新化学家的播客，我们很高兴您能倾听、订阅并收听 Apple Podcasts、Odyssey、Spotify 或任何您收听的地方。欢迎收听播客，在这里好奇心与创新相遇，探索无界限。加入我们，与世界知名的思想领袖交流，包括财政大臣加里梅、罗伯特·兰格博士、格雷格·扎克曼、《华尔街日报》、凯尔·沃姆、福布斯30位30岁以下精英等。透过深入的对话，我们探索科学、领导和目标的变革力量，从突破性研究到改变生活的创新。请注意，本播客的观点代表我和我的嘉宾的观点。此外，这些观点不构成医疗或专业建议，请咨询相关专业人士。我们不承担任何形式的损失。
欢迎收听，今天我们来聊点有意思的，就是几位诺贝尔奖得主，他们亲口讲的那些科学突破的故事。
对，看看那些改变世界的发现背后到底是怎么回事。
是的，你会听到，比如说化学实验里头一个意外的惊喜，或者说在宇宙那么大的尺度上发现的一些惊人的运动。
还有怎么看到原子，甚至细胞里面那些特别复杂的信号传递是怎么运作的。
没错，这些故事里有曲折，有运气，当然也有深刻的洞察力。
是的，关键就是那些啊哈的时刻。我们今天就一起来挖一挖，看看这些突破是怎么发生的，又能给你带来点什么启发。
好，那我们先从化学开始吧。佩克森的故事，他本来想做个实验，合成一个目标分子。
对，目标挺明确的。
结果呢，搞出来一堆黏糊糊的东西，看起来好像没什么用。
对，就是那种没有吸引力的粘稠物，一般人可能就直接扔了。
但他没扔。
他没扔，而且还真从里面就那么一点点分离出了些白色的晶体，产率特别低，可能就0.4%。
哇，那也很了不起了，从废料里。
是，更有意思的是啊，这些晶体几乎不溶解，除非
什么？有什么特殊条件吗？
除非那个溶剂里面还有钠离子。
钠离子，这就很奇怪了啊！溶解性怎么会跟钠离子有关系呢？
对，非常反常。后来他们就研究嘛，用各种光谱方法去看，发现这跟酸碱度什么的都没关系。就是因为钠离子的存在。
那最后搞清楚了吗？结构是什么样的？
搞清楚了，最后用 X 射线衍射一看，这些分子啊是环状的。
环状的。
后来就管它叫冠醚。它那个环状结构正好能像个小笼子一样，把金属离子，比如说钠离子给套进去。
哦，怪不得钠离子能促进溶解。
就是这个道理。所以你看，一个偶然的发现，就因为它没有放过那个反常的现象，结果开创了一个全新的化学领域。
这确实提醒我们，有时候实验里那些不对劲儿的地方，反而是金矿啊。
没错，得留心那些噪音。
好，从分子的微观世界，我们一下子跳到浩瀚的宇宙。这个故事也很有意思。斯穆特团队，他们为了测量宇宙微波背景辐射，就是那个宇宙最古老的光，
对 CMB，
为了测量它上面特别微小的温度差异，竟然用上了U2 高空侦察机。
是的，是的，这个想法本身就很大胆，
听着就很硬核。
那他们测到了吗？
测到了，他们确实测态到了预期的那种非常微弱的温度起伏。但是呢，
对，是有意外。
对，最大的意外是，里面最强的那个信号，就是所谓的偶极各项异性，揭示了一个相当惊人的事实。
什么事实？
就是我们的太阳系，甚至不光太阳系，是咱们整个银河系，正在以一个非常高的速度在宇宙里头漂移。
高速漂移多快？
大概每秒600公里。
每秒600公里。
这比我们想象的宇宙膨胀带来的速度要快得多啊！
对，远超预期。
那是什么意思？是被什么东西拉着走吗？
你问到点子上了。这个发现就意味着，在很远的地方有一个巨大的我们看不见的引力源。
引力源？
对，后来管它叫聚引酸。它就像一个巨大的磁铁，把包括我们银河系、仙女座星系在内的一大片星系，就是所谓的本星系群，都往它那边拉。
哇，这真是感觉宇宙比我们想的还要动态和复杂。
的确如此。这个发现对我们理解宇宙的大尺度结构，还有它是怎么演化的，提出了新的问题会挑战。你看，即使是测量最古老的光，也能反过来告诉我们现在的宇宙正在发生什么，
真是难以置信。好，那我们再把视线拉回来，回到微观世界，看看宾尼西和罗雷尔他们发明扫描吞道显微镜，就是 STM 的故事。听说啊，他们一开始好像没想着要做显微镜。
没错，这个故事也挺有意思的。他们初心的目标其实跟显微镜关系不大。
那他们是怎么走到发明 STM 这一步的呢？
他们当时在研究绝缘层里面的一些导电特性，然后有一次他们观察到一种现象，就是当一个非常尖的金属针尖靠近一个导电表面的时候，即使没有接触，也会有一种微弱的电流流过中间的真空或者绝缘层。
就是量子力学里的那个吞穿效应。
完全正确，就是量子推穿。更关键的是，他们发现这个最穿电流的大小啊，对针尖和表面之间的距离极其敏感，稍微变一点点距离，电流就会发生指数级的变化。
指数级变化。
对，他们一下子就意识到，这个效应可能可以用来做一种全新的探针，来感受物体表面的高低起伏。
哇，这个想法太妙了。
是啊。当然，从想法到实现一个真正能用的显微镜，中间还是有很多挑战的，技术上的，甚至还有一些保密的顾虑。
但他们最终成功了。听说不仅能看到原子是怎么排列的，还能分辨出原子的颜色。这个颜色是指什么？
对，这个颜色啊，其实是个比方。它指的是原子的那种局部的电子特性。就是说，他们不光能获得原子级别的图像，知道原子在哪儿，还能通过调节电压什么的，探测到这个地方的原子它的电子态是怎么样的，
就像给原子做了个电子光谱。
差不多是这个意思。比如他们最早在那个硅111-7x7重构表面上，甚至能分辨出单个的悬挂件的电子态。
太厉害了！这项技术一出来，肯定引起轰动了吧？
那是当然。不过一开始也有人怀疑，觉得是不是计算机模拟出来的假图像。
哈哈，可以理解，毕竟是前所未有的。
是啊，但事实胜于雄辩，这项技术最终被广泛接受，它确实是彻底改变了我们观察，甚至将来操纵物质的方式。它也证明了，有时候一个强大的新工具本身就能开创一个全新的科学领域。
工具的力量确实是巨大的。好，最后我们再往里走一点，深入到细胞内部。杰尔满团队的故事。他们研究的是细胞怎么响应外部的信号，比如激素， 这个问题听起来就很根本。
对，这是一个非常核心的生物学问题。细胞怎么知道外面发生了什么，然后做出反应？他们当时就聚焦在一类叫做肌蛋白的分子上。
肌蛋白，听名字好像是跟能量代谢有点关系。
名字里带 G 是因为它们能结合鸟苷酸，就是 GTP 和 GDP， 它们在信号传递过程中就像一个 switch。
那研究这个肌蛋白难点在哪里？
难点在于这些蛋白质，尤其是他们最早研究的那个 GS 蛋白，在细胞里的含量，非常非常少，而且它们还特别不稳定，很容易失去活性。
那就是提纯非常困难了。
极度困难，需要极大的耐心，还有很高超的生化技术。当他们坚持下来了，
那在研究过程中有什么意外的发现吗？
当然有。首先一个意外就是他们发现这个肌蛋白它不是一个单一的整体，而是有好几个不同的部分，就是所谓的亚基、阿尔法、贝塔、伽马组装起来的一个复合物。这个本身就是个意外发现。
原来是个团队在工作，
可以这么说。而且更复杂的是，后来发现这些亚基本身还会被细胞进行各种很精密的化学修饰。
化学修饰什么意思？比如
说给它加上一些脂肪链之类的东西，像什么肉豆蔻酰化、棕榈下化，听起来有点复杂。
确实。那这些修饰有什么用呢？
用处大了。这些脂肪链就像是给肌蛋白加了毛，能帮助它们正确位置到细胞膜上，或者跟其他蛋白相互作用，对它们发挥正常的信号传递功能是至关重要的。
哇，细胞内部的调控真是精妙到这个程度了。
是的，这揭示了细胞信号调控的复杂性和准确性，远超我们最初的想象。也说明，想要真正理解生命的奥秘往往需要我们深入到分子层面，去关注这些看起来微小的结构和化学变化。
今天听了这几个故事，真是大开眼界。从化学反应里的意外产物，到宇宙尺度的惊人运动，
这都原子级别的微观探索，还有细胞内部那么复杂的机制，
可以看到科学突破的路径真的是非常多样。
确实是这样。有时候真的是靠运气，加上敏锐的观察力，抓住了那个意外。有时候呢，是靠非常精密的测量，揭示了以前不知道的规律，
就像测量CMB那样。
对，还有时候就是因为有了一个强大的新工具，像 STM， 一下子就打开了新世界的大门。
还有就是对复杂生命线上那种锲而不舍的追问。
是的，就像研究鸡蛋白那样，需要耐心和深入。
那么听了这么多过去的故事，最后留给你一个问题，可以思考一下。既然过去的很多重大突破都挺出乎意料的，那下一个可能会改变世界的发现，你觉得最可能隐藏在哪里呢？
会不会就藏在我们现在已经拥有的大量数据里面，只是在等待一种全新的视角或者新的算法去解读它？
这是一种可能。
或者会不会是源于一个我们今天还完全无法想象的新工具，一个能让我们用一种全新的方式去观察这个世界的东西？
值得我们期待，也值得我们继续探索。
欢迎收听，今天我们准备了一串诺贝尔奖得主的演讲精华，想跟你一起从里面挖点干货。
没错，
就是看看这些横跨宇宙学、生物学还有这个工程学的大发现里，最核心的那些想法，还有背后那些挺有意思的故事。
对的，感觉像是走了条近路，快速了解科学前沿。
是这个意思。而且你会发现这些故事里，很多时候科学探索的路子不是直的。对，经常充满了意想不到的转折。有时候是一些让人摸不着头脑的数据，有时候看着像失败的实验，结果呢，反而搞出了革命性的东西。
好，那咱们就先从这个最大的尺度宇宙开始聊。原材料里说发现宇宙在加速膨胀，这个在当时是不是把所有人都给惊着了？
那是相当颠覆。就是1998年初那会儿，研究团队在分析那个特别远的超新星数据，
就是 I 型那个。
对 I 型，分析来分析去，发现，要想解释观测到的那个加速现象就必须得重新用上爱因斯坦那个，他自己后来都觉得不太对的宇宙学常数。
哇，还得把他那个理论捡回来用，
可不是吗？他们自己形容当时的心情，说是介于惊奇和恐惧之间。
惊奇我能理解，恐惧为什么会恐惧？
你想啊，他们反复检查数据，就怕搞错了，把各种干扰因素，比如什么星际尘埃呀什么的，都排除了，结果还是那样，
还是指向加速膨胀。
对，结论就是我们这个宇宙里大概70%都是一种能产生排斥性引力的东西，就是所谓的暗能量。
70%，我的天，这确实够让人害怕的。
后来呢？这个故事还有后续吗？
有更有例子的，他们观测到一颗超级远的 I 型超新星，叫 SN1997FF。它的红移值特别高 ，Z 等于1.7，意思就是它离我们远的不得了，光线都被拉升得很厉害。
那是什么呢？
有意思的地方来了，它看起来比理论预测的要亮了大概60%。
嘿，更远反而更亮，这怎么解释？
这个反而说明啊，宇宙在特别早期的时候，可能是先减速膨胀了一段儿，
先减速，
对，然后才开始加速膨胀。你看，这就让你对宇宙整个演化历史的复杂性是不是又多了点认识？
确实不是一条直线走到黑。
好，那咱们从这个宏大的宇宙一下子跳到微观世界。听说那个绿色荧光蛋白就是 JFP， 现在生物实验室里哪儿都用。它最初的发现也挺神的。
是的，这个故事简直就是无心插柳刘成英的典范。JFP 最早是从一种水母里发现的，
水母会发光的那种。
对，但当时研究人员主要关心的是另一种也会发光的蛋白，叫水母素。JFP 在那个研究里，就是个碍事的污染。
污染？哈哈，这身份可不太光彩。
那他后来怎么就翻身做主了呢？
转折点发生在1992年，有个叫 Roger Tsien 的研究者偶然读到了 Douglas Prusher 的一篇论文，是关于克隆 JFP 基因的。
Prusher 成功了？
没完全成功。Prusher 试着在细菌里表达这个基因，但是效果不好，加上经费也没了，他基本上就快放弃这个项目了。
那太可惜了。
趁烟就联系了 Prusher。Prusher 这人也挺大方，就把他的 DNA 样本分享给了参研。
那参研就成功了？
也没那么容易，挑战还在呢。原始的那个 JFP 啊，它吸收光有2个碰缝，这对于做 FRET 技术来说特别不方便。
FRET 是什么？
简单说就是一种看分子之间靠得多近的技术，需要颜色匹配得很准，有2个碰就不太好用。
那怎么办呢？
他们就做了大量的尝试，搞基因突变，最后发现一个关键的改变就是把第65位那个氨基酸从丝氨酸换成苏氨酸，也就是 S65T 突变。
然后呢？
然后就得到了一个性能超好的 JFP 突变体，只有1个波峰，更亮、更稳定，特别适合用。
哇塞，真不容易！一个差点被扔掉的项目，最后成了照亮细胞内部运作的探照灯。这确实提醒我们，搞科研啊，有时候真的留意那些不起眼的边长链。
对，这种对反常现象的刨根问底，不仅搞出了 GFP， 还揭开了另一个大谜团 ：RNA 干扰，就是 RNAi。RNAi，
基因沉默那个？
没错，最初研究人员是在线虫里研究基因沉默，他们发现往虫子里注射单链的 RNA。不管是正异链还是反异链，效果都不咋地。但更奇怪的是，他们想着是不是 RNA 不纯呢，就去提纯。结果越提纯，那个基因沉默的效果反而越差，甚至没了。
等等，提纯了反而没效果，这完全不合逻辑啊！那他们怎么想的？
正是这个反常现象让他们开了窍。他们意识到起作用的可能根本就不是他们想要的那个单链 RNA。
那是什么？
而是相谱里混着的杂质。后来通过实验证明，这个杂质其实是双链 RNA， 就是 dsRNA。
双链 RNA？
对，就是这个 dsRNA 才能高效而且特别有针对性地引发基因沉默。而且他们还发现，这个沉默过程是伴随着目标信使 RNA，也就是 mRNA 的降解的。
明白了，等于说双链 RNA 是扳机，它能引发一系列反应，把对应的信使 RNA 给干掉。
非常准确。后来进一步的研究就把整个机制搞清楚了。有一种叫 dicer 的酶，像把剪刀，先把长的 dsRNA 切成小片段，然后这些小片段就会被装到一个叫做 RISC 的分子机器。
RISC？
对， RISC 复活物。装进去之后，这个小片段就相当于一个通缉令或者拿行仪，带着 RISC 去找到细胞里跟它序列匹配的那个信使 RNA， 然后咔嚓
把它降解掉，这样相应的蛋白质就合成不了了，基因就沉没了。
这个机制真是太精妙了。所以你看，当初那个让人头疼的提纯失败，反而成了打开一个全新基因调控大门的关键。
的确如此。所以说，在探索未知的时候，遇到那些解释不通的反常，也许恰恰是指路标呢。
这个思路有意思。好，那我们再换个领域，看看化学。电子能谱 ESCA 也叫 XPS， 对吧？这个技术的故事里，是不是也有点意外？
也有。它的开发者 Kai Sigbang， 他最初的目标其实是追求极高的能量分辨率。
分辨率？
对，就是想非常精确地测量原子内部不同电子结合的有多紧，那个能量是多少。他还估算过，说光电子从固体相谱里跑出来，大概能从表面下100I 左右的地方跑出来不损失能量。
100I 也就是10纳米。听起来不算特别浅。那结果怎么样？仪器达到他想要的高分辨率了吗？
不仅达到了，而且还带来了一个一开始让他们挺头疼的醒机。
又是个醒机。
是的，这个仪器表现出一种极端的表面灵敏度。
表面灵敏度多灵敏？
灵敏到什么程度呢？就是你用手指头轻轻碰一下那个相谱表面，再去测，谱图上就会留下非常清晰的指纹污染信号。比如碳元素的那个特征峰形态完全就变了，
用手摸一下就能测出来，那不是给做实验带来很大麻烦？1不小时就污染了。
最初确实觉得是个大麻烦。你想啊，本来想测体相信息的，结果表面一点点东西就干扰这么大。
是啊，
但后来他们就意识到，这种对最表层可能就几个原子层那么深的高度敏感性，反而是这个技术最独特、最牛的地方。
哦，所以缺点变优点了。
没错，于是ESCA XPS 就从一个主要追求体相信息的工具，华丽转身，变成了研究固体材料最表面那层化学成分、化学状态的一个超级强大的武器。你看，一个曾经被认为是副作用或者缺点的特性，最后反而成了它的核心价值。
这真是世事难料啊！本来想挖第三次，结果发现地皮上的草特别值钱。
哈哈，这个比喻挺形象。
好，那最后咱们再穿越回一个多世纪以前，看看无线电通信早期的一个突破。我记得那时候普遍觉得无线电波跟光一样，只能走直线，地球是圆的，那肯定传不远。
对，当时主流观点就是这样，认为地球的曲率会挡住信号，长距离通信基本没戏。但是有一个人不这么想。
谁啊？
就是马可尼 Guglielmo Marconi， 他不信这个邪。
这位大佬他做了什么
？1901年，他在英国南部搞了一个关键实验，选了2个地方，一个在怀特岛，一个在康沃尔郡的利肖角。
这2个地方隔得多远？
大概300公里。
300公里？那按照直线传播理论，这得要多高的天线才能互相看见呀？
理论计算啊，两边的天线至少得超过1600米高。你想想，100多年前，那可能吗？
根本不可能。那他实际用了多高的天线？
实验里2个地方的天线高度都没超过100米。
100米对1600米，这差太远了。那结果呢？信号过去了？
过去了，信号成功的从一头发射，在另一头接收到了。
哇，这怎么可能呢？
这个实验结果就强有力地证明了无线电波它不是严格走直线的，它能够沿着地球表面弯曲传播。虽然当时的理论还解释不了为什么，但这一下子就极大地鼓舞了它。
所以才有了后来更大胆的跨大西洋的通信尝试，对吧？
没错，就是这个看起来不可能的实验给了他巨大的信心。实践直接挑战了当时的理论认知，这说明啊，有时候你得敢于去试那些看起来不可能的事情。
这几个故事听下来真是让人感慨。好了，咱们今天的探索差不多就到这里了。从雨漏的宏大奥秘到细胞里的微小灯塔，从控制基因的复杂开关到材料表面的细微指纹，再到跨越地平线的电波，我们确实看到了科学发现路径的多样性。
是的，希望这些跨越不同领域的知识珍珠啊，能让你感受到科学探索的魅力所在。就像你听到的，很多重大的突破其实 并不是按部就班计划出来的，反而是来自于对那些反常现象的追问，或者是一些意想不到的观察，再或者就是敢于去挑战那些看似板上钉钉的既有认知。
说得太好了。那么最后也留给你一个想法吧。咱们今天生活里身边那些看起来微不足道的细节，或者工作中遇到的一些让人困惑的数据，甚至某个技术上的小毛刺儿，这里面会不会也正悄悄地隐藏着通往下一个重大突破的钥匙呢？
这个想法很有意思。
好了，感谢你的收听，我们下次再见。
再见，
欢迎收听。今天我们来聊点有意思的，深入看看几位诺贝尔奖得主的演讲。
对，我们选了化学、物理、医学好几个领域的。
目标就是从这些顶尖大脑的想法里，帮你挖出那些最让人哇一下的发现，还有他们是怎么想到的。
看看那些灵光一闪的时刻。
准备好了吗？咱们先从田中耕一说起。
好，
他是因为分析蛋白质这类大分子拿奖的，但特别有意思的是，你猜他大学学啥的？
难道不是化学？
不是，电气工程，跟化学可以说是八竿子打不着吧。
哈哈，对，这恰恰就是关键。你想啊，当时最大的坎儿就是怎么把那些又大又脆的蛋白质分子完整的弄到质谱仪里去分析。
对，一不小心就碎了。
是的，田中先生那个技术就是所谓的软激光解析电离。他很聪明，用了一种叫基质的东西。
基质那是干嘛的？
你可以想象成给这些大分子涂一层防晒霜，或者说保护垫。激光先打在基质上，基质吸收能量，然后温和地把大分子推进仪器里。
这样它就不会被激光直接打坏了。
对头，是这个意思。
虽然现在看是重大突破，但你想，一个搞电器的弄出个化学分析法，一开始没多少人觉得这玩意儿靠谱，或者说能有那么大潜力。
确实，跨界思维有时候就是能带来惊喜。好，那我们换个领域，看看医学。这个故事可能更颠覆一些。巴里马歇尔和幽门螺杆菌，
这个很有名。
在他之前，大家不都觉得胃溃疡嘛，肯定是压力大，吃东西不注意造成的。
对，当时的主流观点就是这样，根深蒂固。所以马歇尔和他的合作者罗宾沃伦，他们提出是胃里一种旋形的细菌，就是幽门螺杆菌，
引起了胃炎和溃疡。然后呢，肯定遇到不少阻力吧？
那是相当大。你想想，整个医学界都认为是 A， 你突然跳出来说是 B， 而且还是个以前没听说过的细菌。马歇尔自己都形容那种过有观念，简直像宗教信仰一样。
我好像听说过，他为了证明是不是自己把那个细菌喝下去了。
对，就是这样。这绝对是惊世骇俗了。
哇，真的假的？这胆子也太大了，得对自己多自信呢。
是啊，非常冒险的自我实验，但效果也立竿见影。他很快就出现了胃炎症状，
然后就证明了。
这一下就很有说服力了。最终他们不仅证明了细菌能治病，还弄明白一个事儿，就是这种感染很多都是在小时候发生的。
小时候？
对，很小的时候。所以很多成年病人他不记得自己有过什么急性感染的症状，就解释了为什么以前一直没把细菌和溃疡联系起来。
原来如此，解开了一个大谜团。好，下一个发现可能会让你感觉我们对自己身体的理解可能一直有点死板。
乔治·德黑维西的工作是吧？同位素失踪？
对，你可以想象成给身体里的原子贴上一个小小的追踪标签，看它们都跑哪儿去了。
这个技术带来的发现可以说是革命性的，它揭示了我们的身体其实是一个动态平衡的状态。
动态平衡怎么说？
就是说，你的身体原不是一个固定的，像房子一样的结构，构成你身体的原子和分子，每时每刻都在更新换代。
更新？
对，而且这些原子分子不光是从你吃的食物里来，更多的是你身体内部各个器官之间在不停的交换重组。
内部交换？
没错。打个比方，一个氟酸根离子，它今天可能还在你的肠道黏膜里参与反应，明天就可能跑到血液里，然后进入红细胞，再过2天又跑到肝脏里合成粉脂，最后可能就跑到你的骨头里去了。
哇，等等，你的意思是几年前构成我身体的那些原子，现在大部分 其实都已经不是原来的那些了，都被换掉了。
基本是这样，比例非常高。我们的身体就像一条不断流动的河，旧的水流走了，新的水又补充进来。
天哪，这听起来太不可思议了，完全颠覆了我这个概念的物理基础啊！
的确如此，这个持续流动、不断更新的观念，彻底改变了我们对生命是如何运作的基础认知。
好吧，从身体内部的宏观流动，我们再把镜头拉到更微观的粒子世界。物理学家们也有办法抓住那些我们看不见的基本粒子。
对，戴维·万云兰和沃尔夫冈·保罗的工作就是这方面的代表。
他们是怎么抓粒子的？这个核心思想就是用电场和磁场。你想，离子带电，就可以用电场把它困在一个小空间里，这就是离子径。中子虽然不带电，但它有磁矩，就可以用特殊的磁场，比如磁储存环，把它也束缚住。
就像用无形的笼子或者镊子把它们夹住。
差不多是这个意思。把单个的粒子或者一小撮粒子跟外界隔离开，固定住。
那抓住它们有什么用呢？
用处可大了。一旦你能把单个粒子分离出来，稳定地控制住，你就能对它进行极其精确的测量。比如瓦安兰，他的工作让原子钟的精度提升了好几个数量级，还能在实验室里用几个粒子造出非常小的薛定谔的猫那种量子叠加态，直接检验量子力学的基本原理。
哇！
而保罗的技术呢，甚至可以用来测量像中子这么小的粒子受到的那微乎其微的引力作用。你想想这多惊喜！
难以想象的精度。
对，本质上就是通过这种囚禁和隔离，让人类能以前所未有的清晰度去观察和研究这些物质最基本的组成部分。
明白了。所以我们今天聊的这几个例子，从化学到医学再到物理学，好像都贯穿着一条线索。
是什么？
就是好像都是在挑战那些当时看起来天经地义的观念，不满足于已有的答案。
对，敢于质疑。
然后呢，他们还得想出全新的办法、新的工具或者新的视角，去看见那些以前看不到的东西，不管是胃里的细菌、身体里流动不息的原子，还是被囚禁起来的单个粒子。
没错，说得很好。这些故事恰恰说明了基础科学的进步，很多时候就依赖于这种持续的质疑，还有就是发展新的观察手段。
新的工具带来新的发现。
正是如此。所以最后也留给你一个问题思考一下，在你自己的知识领域或者你的认知体系里，有没有哪些你觉得是板上钉钉的真理或者常识？他们会不会在未来某一天，因为某个新工具的出现，或者一种全新的思考角度而被重新审视，甚至被彻底改编？
Guest list heavy, big names abound, academic and professional leaders, knowledge profound. Gary May, Robert Langer, Golden Sound, podcast lit, wisdom's playground. Conversations rich, interviews bright, from MDs to artists, sharing insight. Chemistry vibe, conversations ignite, steam dreams growing, future so bright. Chancellor May, Sidney Colley in the fold, MK Brown, Sheena Colley stories told, Mike Zott, Robert Langer, wisdom gold, McCall Washington, young minds bold, Dr. Thomas N.G., Todd Golub ignite, Abinaya Marcella, vision bright, Lesel Vincent, Sonola's insight, Michael Evans, cough under spotlight, Sarah Skrabalak, Nigella speak clear, Jake Desmond's vision future near, Mr. Bourne Kuna, Fortingo here, Darissa Ferguson, voices sincere. Guest list heavy, big names abound, academic and professional leaders, knowledge profound. Gary May, Robert Langer, Golden Sound, podcast lit, wisdom's playground. Conversations rich, interviews bright, from MDs to artists, sharing insight. Chemistry vibe, conversations ignite. Steam dreams growing, future so bright. LC Campo, Emery Brown's precision. Lynn Rashay Johnson brings thoughtful vision. Keshav Gavin transclinical mission. Dick Mann and Johansson research ambition. Louis Junghan, Trevor Johnson's art. And Pinder speaking from the heart. David Joshua Ferguson plays his part. Chadwin Hanna, Vincent Miranda start. Greg Zuckerman writing from the Wall Street beat. Cordell Hardy's wisdom research elite. Kyle Woom Forbes list ambition complete. Gabriel Galarza's insights repeat. Guest list heavy, big names abound. Academic and professional leaders, knowledge profound. Gary May, Robert Langer, Golden Sound. Podcast lit, wisdom's playground. Conversations rich, interviews bright, from MDs to artists, sharing insight. Chemistry vibe, conversations ignite. STEAM dreams growing, future so bright. Dev Mandavia, corporate strategy bold. Jenai Burrows, medical stories unfold. Doreen Duncan's vision, Island City mold. Paul Boyer's lecture, stories retold. Omar Sharif Ali, software meta. Economic talks, making minds better. Pranav Dorbala, med school go-getter. Ging Rahim Bia, engineering trendsetter. Savni Kulkarni, Abbott structural heart. Farm D candidate, giving advice to start. Podcast guest list, masterful art. New Chemist podcast, playing art part. Guest list heavy, names renowned. Knowledge deep, esteem, vibes profound. Podcast flowing, innovation sound. Future so bright, keep dreams unbound.
Welcome to the Deep Dive. You've sent us quite the collection of research papers and lecture excerpts.
Yeah.
Everything from the intricate world of ion channels to the history of the Nobel Prize and groundbreaking discoveries in physics, chemistry, biology, even economics.
Yeah.
We've got it all.
Like a greatest hits of science, really spanning decades of breakthroughs across all these different fields.
Yeah, try to tie it all together in this deep dive.
Yeah,
so
I like them to be fun.
We're on the hunt for those aha moments.
Yeah.
The surprising connections that make you see the world a little bit differently.
Exactly. We'll extract the key insights from each source.
Yeah.
But we'll also synthesize, find the common threads, help you grasp the bigger picture.
Yeah, I think one of those aha moments is going to be that story about the scientist who gave himself an ulcer to prove.
Oh, yeah. Talk about dedication.
Talk about dedication.
Indeed.
That's pretty amazing.
And who knew you could mimic the behavior of crystals using just bricks and radio waves?
Bricks and radio waves.
We'll unpack all of that and more.
Okay, I'm intrigued.
But first, to lay the groundwork.
Yeah.
Let's start with something fundamental.
Got it.
Ion channels.
Ion channels, yeah.
This paper you sent on potassium ion channels.
Yeah. Yeah, this is fascinating stuff.
It's mind-blowing how these tiny little gatekeepers in our cells work.
Yeah, and I was reading about it, and it's not just a simple open and shut gate, right? It's much more complex than that.
Described as an ion water queue.
A carefully choreographed dance.
Yeah.
That allows potassium ions to pass through while blocking others.
It's amazing how selective it is.
It is, yeah, very selective.
So how did scientists even figure that out?
Right.
I mean, these channels are way too small to even see.
Well, this is where another Nobel Prize-winning discovery comes.
Right.
X-ray crystallography.
Oh, right.
This allowed researchers to actually map the channel's structure at the atomic level. So seeing this precise arrangement of atoms helps explain how the channel optimizes potassium ion flow. It's pretty incredible how they were able to figure that out.
Mm-hmm.
What I found really cool was the paper's mention of electrostatic repulsion.
Oh, yeah.
Like the potassium ions are pushing each other along.
Right. It seems counterintuitive.
Yeah.
But that repulsion is actually crucial for achieving those super fast conduction rates.
Yeah.
It all comes down to the interplay of multiple factors. The structure, electrostatic forces, even the presence of water molecules.
Yeah.
Very intricate.
This potassium channel study is fascinating on its own.
Yeah.
But it makes me wonder, are there similar mechanisms at play in other types of ion channels?
That's a great question, and it's a question that researchers are actively exploring.
Oh, cool.
There's a whole family of ion channels out there, each with its own specific role and selectivity for different ions. So what we learn about one can often shed light on the workings of others.
It's like a blueprint that they can then use to understand the others.
Yeah, it's a good analogy.
It's a reminder that even the seemingly simple processes in our bodies are incredibly complex.
Incredibly complex and finely tuned.
Finely tuned.
Yeah.
And speaking of breakthroughs that illuminate complexity,
yes,
one of these Nobel lectures describes a game-changing technique called mass spectrometry.
Oh, yes, mass spectrometry.
Mass spectrometry. I remember reading about its use in identifying and analyzing proteins.
Right.
Even those involved in forming ion channels.
Exactly.
So this was
Koichi Tanaka was the Nobel laureate behind this breakthrough.
Yeah.
And he pioneered a way to use lasers to ionize large molecules.
So they're using lasers on molecules now.
It's like shining a light on the molecular world.
Wow.
Allowing us to study these large, complex molecules like never before.
So this laser technique wasn't just an advancement in physics.
No, not at all.
It had a ripple effect across multiple disciplines.
Absolutely.
Including our understanding of those crucial ion channels.
Right.
We were just talking about.
Bringing it all back around. Yeah. Full circle.
It underscores a theme we'll see again and again. Advances in one area can unlock entirely new possibilities in others.
Absolutely. That's the beauty of science.
Yeah.
It's all interconnected.
This is already making me appreciate the interconnectedness of scientific discovery.
That's great to hear.
And that interconnectedness goes beyond just different fields of science.
Right.
Let's take, for example, atomic clocks.
Oh, yes, atomic clocks.
Atomic clocks, the most accurate timekeepers we have. But I've always been a bit fuzzy on how they actually work.
Well, imagine harnessing the incredibly specific frequencies at which atoms absorb and emit light.
I see.
That's the heart of an atomic clock.
Okay.
So you're using the properties of atoms to keep time.
Okay, I'm following so far.
Okay.
Where do lasers come into play?
That's the ingenious part.
Okay.
You see, atoms are constantly jiggling around due to their thermal energy. This introduces uncertainties into those frequency measurements.
Right.
Which makes the clock less accurate.
I see. Yeah.
So how do you get around that?
Right. How do they solve that?
They use lasers.
Oh.
To cool those atoms down.
Wait,
it might seem counterintuitive.
Oh, we're just going to say.
But by carefully tuning lasers to specific frequencies, scientists can essentially chill the atoms.
So lasers, which we usually think of as incredibly hot, can also be used to cool things down.
That's right.
That is mind-blowing.
It's a really cool application of physics.
Yeah.
And it really underscores the importance of challenging our assumptions about what's possible.
Yeah, absolutely.
Because our intuition can often lead us astray.
I never realized something as seemingly abstract as laser cooling could have such a direct impact on something as everyday as timekeeping.
It's a hidden technology that underpins everything from GPS navigation to fundamental scientific research. It's
amazing.
Mind-blowing to think how a discovery in atomic physics can have such far-reaching consequences.
It really can.
Okay, we've gone from the microscopic world of cells to the even tinier world of atoms.
Yeah, what a journey.
I'm excited to see where this deep dive takes us next. But before we jump into a new topic, how about we take a moment to reflect on what we've learned so far?
Sounds good.
What stands out to you from this first part of our journey?
Well, for me, it's that reminder that even the most basic processes in our bodies, like the movement of ions across cell membranes, are just incredibly complex and finely tuned.
They are, and they have to be right.
They do.
Otherwise, things would go haywire.
Yeah.
And the fact that we can understand these processes at the atomic level.
I know, right?
Thanks to techniques like X-ray crystallography.
It's just mind-blowing.
Mind-blowing, yeah.
It really is.
Yeah, and I think it's also fascinating how advancements in one field, like Tanaka's work on mass spectrometry, can have these ripple effects across multiple disciplines, pushing forward our understanding in seemingly unrelated areas.
Exactly. And I think the story of the atomic clocks and laser cooling really illustrates that as well, right?
Absolutely.
Who knew that something so seemingly abstract could have such profound impact on our everyday lives?
Right. You don't think about it, but it's there.
It's just there in the background.
Mm-hmm.
It really speaks to the power of basic research.
It does.
And the importance of exploring the unknown.
Yeah.
Even if the practical applications aren't immediately apparent.
Exactly. Curiosity-driven research is so important.
Yeah.
For pushing the boundaries of knowledge.
Absolutely.
Yeah.
Okay, I'm ready for the next adventure.
All right.
Where to next on our scientific journey?
Well, how about we shift gears from the microscopic to the macroscopic?
Okay.
From the world of atoms and cells to the realm of economics.
Economics, okay.
One of the Nobel lectures we have is from Kenneth Arrow,
right,
who made groundbreaking contributions to general economic equilibrium theory.
Okay. Economics. Now that's a bit of a jump.
It is a bit of a jump.
From ion channels and laser-cooled atoms.
Yeah, we're going from the very small to the very large.
Yeah.
But that's the beauty of this deep dive, right?
Right. We get to
explore all these different areas.
Yeah. What exactly is general economic equilibrium theory?
Right.
And why is it important?
Okay, so it's a complex field.
Yeah.
But in essence, it tries to explain how the actions of individuals, companies, and governments all interact to determine things like the prices of goods and services and how resources are allocated.
So it's like trying to understand how the whole economic machine works.
Yeah, that's a good way to put it.
That sounds incredibly ambitious.
It is very ambitious.
Yeah.
And Arrow's work was particularly important in figuring out under what conditions a competitive market could actually lead to an efficient allocation of resources.
What does that mean, efficient allocation of resources?
No, this is a situation where nobody can be made better off without making someone else worse off.
So that sounds like a pretty ideal scenario.
It does, yeah.
But does it really hold up in the real world?
Right.
Where markets aren't always perfectly competitive?
That's a great question.
Yeah.
It's one that economists continue to debate to this day.
Oh, okay.
Arrow himself acknowledged that real-world markets are often messy.
Yeah, definitely.
With things like monopolies, hidden costs, unequal access to information.
Yeah, lots of things that get in the way.
Lots of complexities.
So his work was more about establishing a theoretical framework than perfectly describing reality.
I think that's a fair assessment, yeah.
Okay, but that doesn't diminish its significance, right?
No, not at all.
I mean, by laying out a rigorous theoretical foundation, Arrow's work gave us a benchmark.
Exactly.
To compare real-world markets
against. A standard to measure against.
Right. It helps us identify where inefficiencies exist.
Right, where things aren't working as well as they could.
Right.
Yeah.
And potentially how to address them.
Exactly.
I can see how that would be valuable for policymakers trying to create a more equitable and functional economy.
Absolutely.
So
his work has had a big impact on policy.
It's applicable to the real world.
Yeah, it's not just theoretical.
Yeah.
It has real-world implications.
And Arrow's contributions went beyond just market economics.
They did.
He also delved into social choice theory, exploring how groups make decisions.
Right, whether it's voting systems or how companies are governed.
So this Nobel lecture isn't just about economics in the traditional sense.
No.
It also offers insights into the dynamics of decision-making.
Absolutely.
In any group setting.
Yeah. In any group setting, yeah.
It's fascinating to see how these seemingly abstract economic theories connect to our everyday lives.
It is, yeah. It brings economics down to earth.
This deep dive is truly showing me how interconnected everything is.
That's what we're hoping to achieve.
And speaking of interconnectedness,
yeah.
Remember those ion channels we discussed earlier?
Oh, yes. The gatekeepers.
Let's talk about another powerful tool that's revolutionized our understanding of them. NMR spectroscopy.
NMR spectroscopy, yes.
Wait, NMR, isn't that the same technology behind MRI scans?
You got it. NMR stands for nuclear magnetic resonance.
Right.
And it's a technique that can be applied in both medicine and fundamental research.
Right.
The Nobel lecture we have is from Kurt Wuthrich, who revolutionized the use of NMR to determine the structures of proteins, including those that make up ion channels.
So, before Wuthrich's work, how did scientists figure out the 3D structure of proteins?
Well, it was a much more laborious process.
I can imagine.
It often involved crystallizing the protein.
Yeah.
Which could be incredibly time-consuming and didn't all work.
Right, and so they had to have huge amounts of the protein.
Exactly.
But Wuthrich
changed the game.
How did he do that?
He figured out how to study proteins in their natural dissolved state.
So you didn't need to crystallize them anymore?
No, which was a huge advantage.
Yeah.
But how does NMR actually work?
Yeah, I mean, it sounds pretty high-tech.
It is high-tech.
Yeah.
It leverages the magnetic properties of atomic nuclei within the protein.
Mm-hmm.
So by analyzing how these nuclei respond to radio waves in a magnetic field, Wuthridge could deduce the protein's 3D structure in solution.
So it's like using radio waves to create a 3D map of the protein.
That's a good analogy.
That's incredible. And this opened up a whole new avenue.
It did, yeah.
Of understanding how proteins function in their natural environment, right?
Precisely. It revolutionized structural biology.
Wow.
Allowing us to study proteins involved in everything from cell signaling.
So it has a wide range of
processes,
applications.
A wide range of applications, yes.
And all this stemmed from a technology initially developed for physics research.
That's right. Another example of that interconnectedness we were talking about earlier.
This deep dive is making me realize how much of science is built upon adapting and refining existing techniques.
It's a constant process of innovation.
Yeah.
Building on what came before.
And sometimes those adaptations lead to breakthroughs in entirely unexpected areas.
Absolutely.
That's exciting.
It is, yeah.
Okay, so we've explored the intricate world of ion channels, a technique for analyzing complex molecules, and even delved into economics and decision-making.
Yeah, quite a range of topics.
Yeah. I can't wait to see what other scientific gems are waiting to be discovered.
There are plenty more to come, I assure you.
Okay.
It's a powerful tool for building molecular rings.
Okay.
Which are like the scaffolding of many organic molecules. The Diels-Alder reaction is a chemist's go-to for creating these rings with elegance and precision.
Now I remember those ring structures were everywhere in organic chemistry. And we have a Nobel lecture on this reaction.
We do.
Oh.
It's from Kurt Alder himself, who shared the Nobel Prize in Chemistry in 1950 with Otto Diels for their discovery and development of this remarkable reaction.
So this lecture takes us back to a time when chemists were developing those fundamental tools for building the complex molecules we rely on today.
Exactly.
I'm curious.
Yeah.
What are some real-world examples of where the Diels-Alder reaction has been used to create important molecules?
Well, one classic example is the synthesis of insecticides, like aldrin and dieldrin, which were widely used in the mid-20th century. The Diels-Alder reaction was key to constructing the complex ring systems in these molecules.
So this isn't just some abstract chemical reaction.
No, not at all.
It's had a real-world impact.
Absolutely.
Helping to control pests and protect crops.
It has, yeah.
And its applications go far beyond insecticides.
Oh, absolutely.
Right.
The Diels-Alder reaction has been used to synthesize countless pharmaceuticals, natural products, and other crucial molecules.
It really highlights how a fundamental understanding of chemical reactions can lead to practical solutions for real-world problems.
It's a great example of that, yeah.
It's like having a molecular toolkit.
I like that analogy.
For building almost anything.
Like a molecular toolkit.
Yeah.
And speaking of toolkits, how about we explore the discovery of a remarkable enzyme called ribonuclease P.
Ribonuclease P.
It plays a crucial role in processing RNA molecules, which we know are essential for carrying genetic information and doing all sorts of jobs in the cell.
Ribonuclease P.
Yeah.
Didn't we already talk about RNA when we discussed Andrew Fire's work on RNA interference?
You did. And this discovery adds another layer to our understanding of RNA. You see, RNA molecules often need to be processed and modified before they can do their jobs properly. And ribonuclease P is a key player in this processing, specifically in the maturation of transfer RNA.
Transfer RNA.
Or tRNA molecules.
Okay, tRNA.
Yeah.
Can you remind me what tRNA molecules do again?
Oh, sure.
I remember they're involved in protein synthesis, but the details are a bit fuzzy.
Well, think of tRNA molecules as the delivery trucks of the cell.
The delivery trucks.
Yeah, they carry specific amino acids to the ribosomes where proteins are assembled.
Ah, right. So ribonuclease P helps to get those tRNA molecules ready for their crucial role in protein synthesis.
Exactly. It's preparing them for their job.
And we have a Nobel lecture on this enzyme.
We do. It's from Sidney Altman.
Sidney Altman.
Who, along with Thomas Sick, received the Nobel Prize in Chemistry in 1989. for their discovery of the catalytic properties of RNA.
Hold on. I thought enzymes were proteins.
Right. That was the thinking at the time.
Yeah.
What do you mean by the catalytic properties of RNA? That's what makes this discovery so revolutionary.
Right.
Up until Altman and Sechs' work, it was a central dogma of biology that only proteins could act as enzymes.
Yeah.
But they turned that dogma on its head.
Okay, so you're telling me that ribonuclease P is made of RNA, not protein.
That's right.
And it acts as an enzyme.
Exactly.
Whoa.
It's called a ribozyme. Yeah, a type of RNA molecule that can catalyze biochemical reactions.
So it's like a protein enzyme, but made of RNA.
Exactly. A very special type of RNA.
Wow, that's incredible.
It is, yeah.
It challenges everything I thought I knew about enzymes.
I know. It's a really mind-bending discovery.
Yeah.
So RNA is not just a passive carrier of genetic information.
Right.
It can also play a very active role in cellular processes.
Right. It's not just a blueprint. It's actually doing things.
It's a worker, too.
Yeah, this is mind-blowing.
Oh, it is, yeah.
And the discovery of ribozymes, like ribonuclease P, has opened up new avenues of research.
It has, yeah.
Right.
Not just in understanding how RNA works, but also in potentially harnessing these ribozymes for therapeutic purposes.
So there could be medical applications of this discovery.
That's the hope. Yeah.
It makes you wonder what other surprises RNA has in store for us.
It does. There's still so much we don't know about RNA.
Yeah.
It's a fascinating molecule.
And speaking of surprises,
how
about we delve into the groundbreaking work of Stanford Moore and William Stein on the structure and function of ribonuclease.
Ribonuclease, okay.
A protein enzyme that degrades RNA molecules.
All right, so now we're looking at the other side of the coin.
Ribonuclease.
Yeah.
Wait a minute. Isn't that the opposite of what ribonuclease P does?
That's a great observation.
Okay.
While ribonuclease P is involved in processing and maturing RNA molecules, ribonuclease, often called RNase, is responsible for breaking them down.
So we're exploring two sides of the same coin.
Exactly.
The synthesis and degradation of RNA molecules.
It's all about balance, right?
Right.
You need both processes to maintain proper cellular function.
And Moore and Stein's work on RNAs was groundbreaking for several reasons.
It was.
Right.
They were among the first to determine the complete amino acid sequence of a protein.
The complete amino acid sequence.
Yeah, essentially providing a blueprint of its molecular structure.
So their work was crucial in establishing the link between a protein's amino acid sequence, its building blocks, and its three-dimensional structure.
And ultimately its function. Yeah, it's all connected.
And their meticulous research on RNase's mechanism of action revealed how this enzyme specifically recognizes and cuts RNA molecules.
Right, they really delved into the details of how it works,
providing valuable insights into the principles of how enzymes work.
It was a major contribution to our understanding of enzymology.
This deep dive is reminding me that to truly understand biological processes, we need to look at both sides of the equation.
Exactly.
The building up and the breaking down of molecules.
It's all about maintaining that delicate balance.
It's all about delicate balance.
Yep.
Yeah.
And speaking of balance, how about we shift our attention to the work of Earl Sutherland, who discovered a key molecule involved in cellular signaling.
Okay.
Cyclic AMP.
Cyclic AMP.
Or AMP, as it's often called.
Cyclic AMP. That rings a bell in my biology class, but I'm a bit rusty on the details.
Okay, well, think of it as a cellular messenger.
Okay.
Relaying signals from outside the cell to inside the cell.
A cellular messenger.
It helps cells respond to hormones and other external cues.
I see.
Ensuring they're doing the right thing at the right time.
So it's like a molecular communication system within the cell.
That's a good way to put it, yeah.
What makes cyclic AMP or KMP
so special?
Well, Sutherland's discovery of KMP was groundbreaking.
Okay.
Because it revealed a fundamental mechanism by which cells communicate and respond to their environment.
So it's like a universal language that cells use to talk to each other.
Yeah, a way for them to send and receive messages.
Wow. That's a pretty big deal. And we have a Nobel lecture on this discovery.
We do. It's from Earl Sutherland himself.
Okay.
Who won the Nobel Prize in Physiology or Medicine in 1971.
Right.
For his discoveries concerning the mechanisms of how hormones work.
Right.
Specifically, his work on KMP.
So this lecture takes us back to a time when scientists were just starting to unravel those complex communication networks.
It does, yeah.
Within cells.
It was a time of great discovery in cell biology.
It must have been like deciphering a secret code.
It was very much like that, trying to figure out how cells talk to each other.
Yeah. What were some of Sutherland's key findings about how this cellular messaging system works? Well, one of the crucial findings was that CAMP is generated from ATP, the cell's primary energy currency, by an enzyme called adenyl cyclase.
Adenyl cyclase. Okay.
And this enzyme gets activated by various hormones and neurotransmitters, leading to a surge in CAMP levels within the cell.
So it's like a cascade of events. A signal from outside the cell triggers a series of molecular changes inside the cell.
Exactly.
Ultimately leading to a specific response.
That's how signaling pathways work.
What kind of responses are we talking about?
Well, everything from changes in metabolism to alterations in gene expression.
Right.
Sutherland's work revealed that CANAM activates a key enzyme called protein kinase.
Protein kinase A.
Which in turn modifies and regulates a whole slew of target proteins.
Oh, wow.
Leading to a wide range of cellular responses.
This deep dive is highlighting the incredible complexity of cellular signaling.
It is, yeah.
It's amazing how our cells are constantly receiving and interpreting signals from their environment.
They're busy.
And orchestrating a symphony of responses.
It's like a beautifully orchestrated dance happening inside every cell.
To maintain balance and function.
Exactly.
And speaking of revolutionary technologies, let's shift gears back to the realm of electronics and delve into William Shockley's Nobel lecture on transistor technology.
Transistor technology, yes.
Transistor technology.
Yeah.
Didn't we already discuss transistors when we talked about John Bardine's Nobel lecture?
Here we did.
Okay, so...
But Shockley's lecture offers a different perspective.
A different perspective.
While we explored the invention of the transistor and the scientific principles behind it, Shockley focuses more on the evolution and impact of transistor technology.
Okay.
Particularly its contributions to physics.
Ah, okay. So we're going beyond just the invention itself and looking at how transistor technology has shaped our understanding of the physical world.
Exactly.
That's an interesting angle.
It is. Shockley's lecture highlights how the development of transistors led to new insights into the behavior of electrons in solids.
Okay.
Particularly in semiconductors.
It's fascinating how a technology that revolutionized electronics also ended up deepening our understanding of the fundamental properties of matter.
It's a great example of how technology can drive scientific discovery and vice versa.
And Shockley's work was instrumental in establishing the field of solid-state physics.
Absolutely.
Which underpins so much of our modern technology.
It does, yeah. It's the foundation for so many things.
His lecture also mentions the concept of deathnium.
Oh, yes. Deathnium.
Right. It sounds a bit ominous.
It does, doesn't it?
Yeah.
But it's actually a term he coined to describe a type of imperfection in a crystal lattice that can trap electrons, affecting the conductivity of the material.
So it's like a tiny defect in the otherwise perfect arrangement of atoms in a crystal.
Yeah, that's a good way to think about it.
That can disrupt the flow of electrons.
Exactly.
Okay, so it's not...
It's a bit of a whimsical term.
Yeah, it's not a scary...
It's for a serious concept.
Yes, it sounds.
Yeah.
But it's important.
But it is an important concept because these imperfections can have a big impact on how semiconductors behave.
Right.
Shockley's insights into the role of imperfections in semiconductors were crucial for improving transistor performance and developing new semiconductor materials.
This is a great example of how understanding the nuances of a material, even its imperfections, can be crucial for harnessing its full potential. It's about understanding how things work at the most fundamental level.
Exactly. It's all about the details.
And speaking of fundamental discoveries, how about we venture into the world of nuclear physics?
All right, let's do it.
And explore Maria Guppert-Mayer's Nobel lecture on the nuclear shell model.
The nuclear shell model.
The nuclear shell model. Right. I'm not familiar with that one.
Okay, well...
Can you give me a crash course?
Imagine the nucleus of an atom.
Okay.
That tiny, dense core made up of protons and neutrons.
Right, right.
The nuclear shell model is a way of understanding how those protons and neutrons are arranged within the nucleus.
I see.
It proposes that they occupy distinct energy levels, or shells.
Oh.
Much like electrons occupy shells in an atom.
So We're talking about the internal structure of atomic nuclei.
Exactly.
How those fundamental building blocks are organized.
Yes. It's a way of understanding the architecture of the nucleus.
Okay.
And the nuclear shell model was a breakthrough in understanding the behavior of nuclei.
Right.
It successfully explained many of the observed properties of nuclei.
Such as
their stability, their spin, and their magnetic moments.
So things that had puzzled scientists?
Things that had puzzled scientists for a long time.
For a long time.
Yeah, it was a major step forward.
And we have a Nobel lecture on this model.
We do.
Okay.
It's from Maria Goeppert-Mayer herself.
Maria Goeppert-Mayer.
Who shared the Nobel Prize in Physics in 1963 with J. Hans D. Jensen for their independent development of the nuclear shell model.
This deep dive is not only shedding light on incredible scientific discoveries, but also celebrating some of the brilliant minds behind them.
It is, it's a celebration of human ingenuity and curiosity.
Yeah, it's really inspiring.
It is.
So back to the nuclear shell model.
Okay.
What were some of the key features of this model that Goeppert-Mayer emphasized in her lecture?
Well, one of the most important features was the introduction of a strong spin-orbit interaction.
Spin-orbit interaction.
Yeah, this means that the energy levels of protons and neutrons are affected by their intrinsic angular momentum.
Right. So it's not just about the spatial arrangement of those protons and neutrons.
No, it's not just about where they are.
Within the nucleus.
Their internal spin also plays a role in determining their energy levels.
It's incredible how, even at the subatomic level, there are these intricate layers of complexity and interplay between different forces.
Yeah, it's a very intricate dance happening at the heart of matter.
And the nuclear shell model, well, a simplification of reality.
Right, it's a model.
Provides a powerful framework.
But it's a very useful model for understanding the behavior of atomic nuclei.
This deep dive is really highlighting the power of theoretical models in science.
Absolutely. Models are essential tools for understanding complex systems.
They provide a lens through which we can view and make sense of complex phenomena.
Yeah, they help us to simplify.
Even if they can't capture every nuance.
And make sense of the complexity.
Right, right.
Yeah.
Okay, where does this scientific adventure take us next?
How about we explore the work of Wolfgang Paul? Who developed a technique for trapping charged and neutral particles.
Trapping particles.
Using electromagnetic fields.
With electromagnetic fields. That sounds pretty futuristic.
It does sound futuristic, doesn't it?
Yeah.
But it's a real technique that has revolutionized our ability to study and manipulate particles at the atomic level.
Can you paint a picture of what that looks like?
Sure.
And why it's so important?
So imagine creating a tiny invisible cage made of electric and magnetic fields to trap individual particles.
Wow.
Like ions.
So they're using...
These devices are called ion traps.
Ion traps.
Yeah, and they have revolutionized our ability to study and manipulate particles.
Sounds like something straight out of science fiction.
It does. It's pretty amazing technology.
Yeah.
But it's real.
Yeah.
And it's led to some incredible advancements in our understanding of the universe.
And we have a Nobel lecture from Wolfgang Paul himself.
We do. Who shared the Nobel Prize in Physics in 1989.
Wow.
With Norman Ramsey and Hans Dehmelt. For their work on developing techniques for trapping and studying atoms and ions.
So this lecture delves into the world of precision measurement?
It does, yeah.
And control at the atomic scale?
It's all about manipulating and measuring things at the tiniest level.
I'm curious, what are some specific examples of how ion traps have been used to push the boundaries of scientific knowledge?
Well, one of the most remarkable applications is in atomic clocks.
Atomic clocks?
The most accurate timekeepers ever invented.
Right. I remember you were talking about those earlier.
Ion traps are used to confine and cool ions to extremely low temperatures, slowing their motion, and allowing for incredibly precise measurements of their atomic transitions.
So they're basically freezing them in place.
Essentially, yes.
So they can measure them more accurately.
Yeah. It's all about reducing those uncertainties we talked about earlier.
Wow. So Paul's work on ion traps has indirectly contributed.
It has, yeah.
To the accuracy of our GPS systems and other technologies that rely on precise timekeeping.
Absolutely.
Pretty mind-blowing.
It's pretty amazing.
And the applications of ion traps extend far beyond atomic clocks.
Oh, absolutely.
Right.
They're used in mass spectrometry to identify and analyze molecules.
Right.
In quantum computing to manipulate qubits.
Qubits?
Yeah.
Okay.
And even in biological research to study the structure and function of proteins.
So they have a wide range of applications.
Yes, they do, yeah.
It seems like ion traps are an incredibly versatile tool.
They are.
With applications across a wide range of scientific disciplines.
And it highlights another recurring theme in this deep dive.
Okay.
The interconnectedness of scientific knowledge.
Right.
And how advancements in one field can have a ripple effect across many others.
Yeah, absolutely. Okay, I'm ready for another adventure.
All right, let's go.
Where will this exploration take us next?
Well, how about we shift our focus from the microscopic world of atoms and ions to the fascinating realm of vision and neurobiology.
Okay, vision.
Mm-hmm.
We have a Nobel lecture from H. Keffer Hartline.
H. Keffer Hartline.
On visual receptors and retinal interaction.
Retinal interaction.
Visual receptors and retinal interaction.
So we're diving into the workings of the eye and the brain.
I've always been fascinated by how our senses work.
Yeah, it's amazing.
Especially vision.
It's how we perceive the world around us.
Yes.
It's a very complex process.
It's a very complex process.
Hartline's work focused on understanding how our eyes detect light signals and how those signals are processed by the nervous system to create our sense of vision.
And we have a Nobel lecture on this very topic.
We do. It's from H. Keffer Hartline himself, who shared the Nobel Prize in Physiology or Medicine in 1967 with Ragnar Granit and George Wald. Further discoveries concerning the primary physiological and chemical visual processes in the eye.
It's incredible to think
It is.
we can understand something as complex as vision at such a fundamental level. What was Hartline's approach to studying vision?
Well, his research focused on the horseshoe crab.
The horseshoe crab.
Which has a relatively simple visual system compared to ours.
Oh, I see.
This made it an ideal model.
They're using a simpler system.
For studying the basic principles of vision.
To understand the more complex one.
Exactly.
So, by studying a simpler system, he was able to gain insights into the fundamental mechanisms of vision.
That's the idea.
That apply across species.
That's right. It's a common approach in science.
Yeah, it makes sense.
Studying a simpler system to gain insights into more complex ones.
What were some of the key insights
that
Hartline's research on the horseshoe crab revealed?
Well, one of the key findings was that individual photoreceptor cells in the eye, the cells that actually detect light, respond to light with a burst of electrical signals.
So each photoreceptor cell acts like a tiny light sensor.
Yeah, that's a good analogy.
Sending signals to the brain when it detects light.
Exactly. It's converting light into electrical signals.
But it's not just about individual cells working in isolation.
No, it's not just about individual cells.
Right.
Hartline's research also revealed that these photoreceptor cells are interconnected in a way that allows them to influence each other's responses.
Ah, so the signals from individual photoreceptors aren't just sent directly to the brain.
No, there's more to it than that.
There's some processing and integration happening
exactly
within the retina itself.
That light-sensitive layer at the back of the eye.
Right, right.
Hartline discovered a phenomenon called lateral inhibition.
Lateral inhibition.
Where the activity of one photoreceptor cell can actually inhibit the activity of its neighbors.
So one cell can tell its neighbor to quiet down.
Essentially, yes.
That's so interesting.
It is, yeah.
Why would they do that?
Well, it might seem counterintuitive, but it actually enhances contrast and sharpens the visual image.
Oh, I see.
So it's a way of refining the visual information before it even gets to the brain.
So our eyes are doing more than just passively detecting light.
They are, yeah.
There's a whole lot of signal processing going on.
It's a very active process in the retina to refine and enhance the visual information before it even gets to the brain.
It's amazing how much complexity is packed into something as seemingly simple as seeing.
It really is. Vision is a marvel of biology and physics.
And speaking of amazing phenomena, how about we explore Ivar Jeever's Nobel lecture on the phenomenon of tunneling in superconductors.
Tunneling in superconductors.
Tunneling in superconductors.
All right.
Now that sounds like we're venturing into the realm of quantum mechanics again.
It does, yeah.
But first, can you remind me what superconductors are?
Okay.
I know they have something to do with electricity, but the details are a bit fuzzy.
Well, think of superconductors as materials
Okay.
that can conduct electricity
Right.
with zero resistance.
With zero resistance.
So it's like a superhighway for electrons.
Wow.
Where they can flow
Yeah.
without any loss of energy.
Wow, that's incredible.
It is, yeah. It's a very special property.
But what does tunneling have to do with superconductors?
That's okay. So tunneling is a quantum mechanical phenomenon
Right.
where particles like electrons can pass through barriers
Right.
that would be impossible for them to cross
Yeah,
according to classical physics.
Right. It's like they can magically teleport through walls.
It's a very strange phenomenon,
It's very strange.
but it's a real phenomenon.
Quantum mechanics never ceases to amaze me.
It's full of surprises.
So how does tunneling manifest in superconductors?
Well, Jagever's work demonstrated that electrons can tunnel through thin insulating layers, separating two superconductors. This seemingly impossible feat provided crucial evidence for the theory of superconductivity.
So it helped to confirm the theory.
It did, yeah. It was a key piece of the puzzle.
And we have a Nobel lecture on this very discovery.
We do. It's from Ivar Gier himself, who shared the Nobel Prize in Physics in 1973 with Brian Josephson and Leo Asaki for their experimental discoveries regarding tunneling phenomena in semiconductors and superconductors.
So this lecture takes us back to a time when physicists were grappling with the mind-boggling world of quantum mechanics.
It does, yeah.
And its implications for material science.
It was a time of great excitement and discovery in the field of condensed matter physics.
What were some of the key implications of Javier's discovery? Did it have any practical applications?
Well, his work not only confirmed the theory of superconductivity, but it also paved the way for the development of incredibly sensitive devices, such as superconducting quantum interference devices.
Superconducting quantum interference devices.
Or SQUIDs.
SQUIDs, okay.
These devices are used in a wide range of applications.
So
from medical imaging to geological exploration.
So this fundamental research into a rather esoteric phenomena ended up having a tangible impact on technology.
It did, yeah.
And our ability to study the world around us.
Sometimes the most unexpected discoveries come from exploring.
That's pretty amazing.
Those seemingly bizarre corners of the universe.
It is.
It underscores the importance of curiosity-driven research.
Yeah. Okay, I'm ready for another scientific adventure. What's next on our agenda?
How about we shift gears from the world of physics to the realm of medicine.
Medicine.
And explore Barry Marshall's Nobel lecture on the discovery of Helicobacter pylori.
Helicobacter pylori.
And its role in gastric ulcers.
Helicobacter pylori.
Yeah.
The bacterium that causes stomach ulcers. I remember learning about that in biology class.
Yeah, it's a fascinating story.
But I'm curious to hear the story behind its discovery.
It was a rather controversial finding at the time.
It seems like it was a rather controversial finding at the time.
It was, yeah.
Yeah.
For decades, the medical community was convinced that stomach ulcers were caused by stress.
Stress.
The spicy food, excess acid production.
Right, the old conventional wisdom.
The old thinking.
But Barry Marshall and his colleague Robin Warren had a different idea.
They did.
They suspected a bacterium was the culprit. And they were right. I vaguely remember reading that Marshall even infected himself with H. pylori to prove his point.
He did. It's a famous story in the history of medicine.
Talk about dedication to science.
Yeah.
But before we get into that, you mentioned we have a Nobel lecture on this discovery.
We do. It's from Barry Marshall himself, who shared the Nobel Prize in Physiology or Medicine in 2005 with Robin Warren. For their discovery of Helicobacter pylori and its role in gastritis and peptic ulcer disease.
So this lecture takes us back to a time when the medical community was pretty skeptical.
It does.
Of the idea that bacteria could be the root cause of ulcers.
It was a paradigm shift.
What kind of challenges did Marshall and Warren face?
Oh, it was an uphill battle.
In trying to convince the world of their discovery.
Their initial findings were met with resistance and skepticism. Many in the medical community who were wedded to the old dogma about stress and lifestyle factors being the main culprits behind ulcers.
So how do they ultimately convince the skeptics?
Well, they presented compelling evidence from their research showing a clear link between H. pylori infection and the development of ulcers.
But perhaps the most convincing piece of evidence came from Marshall himself.
Yeah, that's right.
Right.
He deliberately infected himself with H. pylori. Developed gastritis and then successfully treated it with antibiotics. Talk about a self-experiment.
Talk about putting your money where your mouth is. It was a bold move that ultimately helped change the course of medical history.
It was a game changer.
Yeah.
For our understanding of ulcers.
What were some of the key insights into H. pylori and its role in ulcers that Marshall's research revealed?
Well, one of the key findings was that H. pylori is incredibly well adapted to survive in the harsh acidic environment of the stomach.
It's got to be a tough place to live.
It is, yeah. It produces an enzyme called urease, which neutralizes stomach acid, creating a more hospitable niche for itself.
So it's like this bacterium brings its own antacid to the party.
That's a good way to put it.
What a clever survival strategy.
And Marshall's research showed that this H. pylori infection triggers an inflammatory response in the stomach lining, leading to gastritis.
Gastritis?
Which can develop into ulcers in some individuals.
This deep dive is highlighting the importance of challenging dogma in science.
It is, yeah.
It's a reminder that our understanding of the world is constantly evolving.
Absolutely.
And that sometimes the most groundbreaking discoveries come from questioning the status quo.
That's what drives progress.
Couldn't agree more.
Yeah.
Marshall and Warren's willingness to challenge conventional wisdom ultimately led to a paradigm shift in our understanding of ulcers.
It revolutionized the treatment of ulcers.
Transforming their treatment and improving the lives of countless people.
Countless people, yeah. It's a great example of how scientific discoveries can have a real impact on human health.
It's an inspiring story.
It is.
Of scientific tenacity.
Yeah.
And the power of questioning assumptions.
A great reminder to never take things for granted.
Yeah.
Always be questioning.
Indeed. Now, speaking of paradigm shifts.
Okay.
How about we explore the work of Elias James Corey?
Elias James Corey.
Who developed a revolutionary approach to organic synthesis called retrosynthetic analysis.
Retrosynthetic analysis. Okay.
Retrosynthetic analysis.
That sounds like a strategic and systematic approach to planning chemical reactions.
Yeah.
I remember organic synthesis being presented as a bit of a puzzle.
Yeah, it was a puzzle.
With chemists trying to figure out how to assemble complex molecules from simply starting materials.
Like a molecular jigsaw puzzle.
It is, yeah. And before Corey's work, organic synthesis often involved a lot of trial and error. With chemists trying different reactions and hoping for the best.
Hoping for the best, yeah.
But Corey's retrosynthetic analysis brought order to the chaos.
And we have a Nobel lecture on this approach.
We do. It's from Elias James Corey himself.
Okay.
Who won the Nobel Prize in Chemistry in 1990. For his development of the theory and methodology of organic synthesis. Particularly his work on retrosynthetic analysis.
So this lecture takes us into the world of molecular design, where chemists use their knowledge of chemical reactions to strategize and execute the synthesis of complex molecules.
Like a molecular architect.
It is like an architect.
Yeah, planning out how to build these complex structures.
So Corey's lecture lays out the principles
of retrosynthetic analysis.
Okay?
You're working fast.
Which involves working backward from the target molecule.
So instead of starting from the beginning,
you start from the end.
They start from the end.
And work your way back. You break it down into simpler and simpler precursors. Until you arrive at readily available starting materials.
So it's like reverse engineering a molecule.
That's a great analogy.
You start with the end product and then deconstruct it to figure out the best way to build it from scratch.
Exactly. It's a very clever approach.
That's really cool.
And Corey's lecture highlights the power of this approach in simplifying complex synthetic problems.
Enabling chemists to create an astonishing array of molecules. From natural products to new drugs.
What are some specific examples of how retrosynthetic analysis has been used to create molecules that have had a real impact?
Well, one of the most notable examples is Corey's own synthesis of prostaglandins.
Prostaglandins.
These are hormone-like molecules involved in a wide range of physiological processes. From inflammation to blood clotting. Retrosynthetic analysis was key to planning and executing the synthesis of these incredible incredibly complex molecules.
So this isn't just some theoretical exercise.
No, not at all.
It's a powerful tool that has led to the creation of molecules that have improved human health and well-being.
Absolutely. It's a great example of how chemistry can be used to solve real-world problems.
This deep dive is showcasing the amazing ingenuity of scientists in tackling complex challenges.
Yeah, it is, yeah.
Whether it's developing powerful techniques like NMR spectroscopy or creating systematic approaches.
Like retrosynthetic analysis. It's all driven by a desire to understand and manipulate the world around us.
I couldn't agree more. And speaking of ingenuity, how about we delve into the work of Paul Lauterbur and Sir Peter Mansfield, who revolutionized medical imaging with the development of magnetic resonance imaging.
MRI.
Or MRI.
MRI.
Now, that's a technology I'm familiar with.
It's a very powerful technology.
It's incredible how we can create detailed images of the inside of the body without using harmful radiation.
It's amazing, isn't it?
It truly is. And Lauterbur and Mansfield's work on MRI was groundbreaking.
It was a major breakthrough in medical imaging.
They combined principles from physics, chemistry, and engineering.
A truly interdisciplinary effort
to create a technique that has revolutionized medical diagnosis and research.
It's had a huge impact on medicine.
And we have a Nobel lecture on this revolutionary technology.
We do. It's from Paul Lauterbur. Who shared the Nobel Prize in Physiology or Medicine in 2003. With Sir Peter Mansfield. For their discoveries concerning magnetic resonance imaging.
So this lecture takes us back to a time.
It does.
When medical imaging was undergoing a major transformation.
It was a time of great innovation in the field.
With new techniques like CT scans.
CT scans, ultrasound.
And ultrasound also emerging.
Lots of new ways to see it inside the body.
What were some of the key insights that led to the development of MRI? I know it involves magnetism, but I'm a bit fuzzy on the details.
Well, one of the key insights was Lauterbur's realization that by varying the magnetic field across a sample, it was possible to encode spatial information into the NMR signals.
So by creating a sort of magnetic map,
essentially, yes,
different regions of the body would emit distinct signals that could be used to reconstruct an image.
Exactly.
It's incredible how they were able to bridge the gap between physics and medicine. Using principles of magnetism and nuclear resonance to visualize the human body in such detail.
It was a truly remarkable achievement.
It's a testament to the power of interdisciplinary research.
I agree.
And the amazing things that can happen when scientists from different fields come together to tackle a challenge.
Breaking down those barriers between disciplines.
This deep dive is highlighting that interdisciplinary approach time and time again.
Yeah, it's a recurring theme.
It's a reminder that some of the most groundbreaking discoveries come from bridging the gaps between traditional disciplines.
It's at those intersections that the real magic happens.
Okay, where to next on our scientific exploration?
How about we return to the world of economics?
Back to economics,
and explore Lawrence Klein's Nobel lecture on the development of econometric models.
Econometric models.
For analyzing the global economy.
Econometric models.
The mathematical representations of economic systems. Using statistical methods to analyze data and predict future trends.
So it's like trying to capture the complex workings of the economy.
Yeah, it's a very ambitious undertaking in a mathematical equation. That seems incredibly ambitious. It is very ambitious.
And Klein's work was pioneering because he developed large-scale econometric models that could be used to analyze the entire global economy.
Not just individual countries or sectors. He was trying to model the entire system.
And we have a Nobel lecture from him on this topic.
We do. It's from Lawrence Klein himself. Who received the Nobel Prize in Economics in 1980. For his work on creating these econometric models and applying them to analyze things like economic fluctuations and the impact of different economic policies.
So this lecture takes us back to a time when economists were trying to grapple with the growing complexity.
The global economy was becoming increasingly interconnected.
Of the interconnected global economy.
And Klein's work was at the forefront of trying to understand how to model and analyze that complexity.
What were some of the key aspects of Klein's econometric models
that
he emphasized in his lecture.
Well, one of the key features was their comprehensiveness.
Comprehensiveness.
His models incorporated data on a wide range of economic variables, from consumption and investment to government spending and international trade.
So his models aim to capture the interconnectedness of the global economy.
Exactly.
Considering all the different factors.
He recognized that trying to isolate individual parts of the economy wouldn't give you an accurate picture of how it functions as a whole.
That makes sense.
And another key feature was their dynamic nature.
Dynamic nature.
Yeah, Klein's models didn't just provide a static snapshot of the economy. They aimed to simulate how it might evolve over time.
So they weren't just a picture in time.
No, they were trying to...
A model that could...
predict how the economy might change in response to different factors.
This deep dive is highlighting the power of mathematical modeling in science.
It is, yeah.
Whether we're talking about the nuclear shell model or econometric models, they give us frameworks for understanding complex systems.
Models are essential tools for scientists in many different fields.
Even if they can't perfectly replicate every aspect of reality.
Of course, models are simplifications, but they can be very powerful simplifications.
They're tools.
They help us to understand the essential features of complex systems.
Absolutely.
Yeah.
Okay, I'm excited to see where this deep dive takes us next.
How about we return to the realm of chemistry?
Back to chemistry.
Yeah. Okay. And explore Irving Langmuir's Nobel lecture on surface chemistry.
Surface chemistry. Surface chemistry.
That sounds like the branch of chemistry.
That's what it sounds like.
That deals with the interactions between molecules at surfaces. You got it. And it's a crucial area of study because those surface interactions are essential for a vast range of phenomena, from how catalysts work to the corrosion of metals to the behavior of biological membranes.
It's a lot of stuff.
Langmuir's work laid the foundation for our understanding of this important field.
And we have a Nobel lecture from him on this very topic.
We do. It's from Irving Langmuir himself.
Irving Langmuir,
who won the Nobel Prize in Chemistry in 1932 for his discoveries and investigations in surface chemistry.
So this lecture takes us back to a time when chemists were just beginning to appreciate the unique properties and behavior of molecules at surfaces.
It was a new frontier in chemistry.
What were some of the key principles of surface chemistry? That Langmuir highlighted in his lecture.
Well, one of the key principles was his model of monolayer adsorption.
Monolayer adsorption.
Which proposed that molecules adsorb to surfaces in a single layer. Each molecule occupying a specific site on the surface.
So it's like a molecular puzzle?
Yeah, that's a good analogy.
With each molecule snapping into a specific spot on the surface.
It's a very ordered arrangement.
And Langmuir's work also revealed the importance of surface tension.
Surface tension.
The force that causes liquids to minimize their surface area.
Right. That's why water droplets form beads. It's trying to minimize its surface area.
In governing the behavior of molecules at interfaces.
It's a very important concept in surface chemistry.
This deep dive is highlighting the importance of understanding how things work. At the most fundamental level.
Yeah, that's what science is all about.
Langmuir's work on surface chemistry showed that the behavior of molecules at surfaces can be vastly different from their behavior in the bulk of a liquid.
They behave differently at the interface than they do in the bulk.
This has huge implications for understanding.
It does.
Chemical and biological processes.
It affects everything from how reactions occur to how cells interact with each other.
And his insights have had a profound impact on various fields, from material science
to nanotechnology to biomedicine.
Okay, I'm ready for another scientific adventure. What's up next?
How about we delve into the world of biochemistry and explore Carl and Gertie Corey's Nobel lecture.
Carl and Gertie Corey.
On discovery and characterization of the enzyme polysaccharide phosphorylase.
Polysaccharide phosphorylase.
Okay, are you ready for another mouthful of a name?
Yeah, I think I can handle it.
All right.
Polysaccharide phosphorylase.
That sounds like it's involved in breaking down carbohydrates.
It does, doesn't it?
You're on the right track. Polysaccharide phosphorylase.
Okay.
Or phosphorylase for short.
Phosphorylase.
Is a key enzyme involved in the breakdown and synthesis of glycogen.
Glycogen.
A complex carbohydrate that serves as a storage form of glucose in animals.
Okay, so we're talking about the enzyme that helps our bodies store and release energy.
Yeah, it's all about energy balance.
From carbohydrates, ensuring a steady supply of fuel for our cells.
It's a key player in metabolism.
And we have a Nobel lecture on this enzyme.
We do. It's from Carl and Gertie Corey.
Carl and Gertie Corey.
Who shared the Nobel Prize in Physiology or Medicine in 1947.
Right.
With Bernardo Houssay.
Okay.
For their discoveries relating to the catalytic conversion of glycogen.
Right.
Specifically their work on phosphorylase.
I remember reading about Gertie Corey. She was the first American woman to win a Nobel Prize in science.
That's right. She and her husband, Carl, were a remarkable scientific duo.
Yeah, I've heard of them.
Making groundbreaking contributions to our understanding of biochemistry and metabolism.
What were some of their key findings about phosphorylase?
Okay.
And its role in glycogen metabolism?
Well, one of their key findings was that phosphorylase catalyzes the breakdown of glycogen into glucose 1-phosphate.
Glucose 1-phosphate.
Yeah, a crucial intermediate
in glucose metabolism.
And this process is reversible, right?
It is, yes.
All right.
So phosphorylase can also help build glycogen back up from glucose 1-phosphate.
So phosphorylase acts like a molecular switch.
That's a good analogy.
Controlling the flow of glucose into and out of storage.
It's regulating the balance
Yeah.
between storage and release.
Depending on the body's needs.
And the chorus also discovered that phosphorylase exists in two forms.
Two forms?
An active form and an inactive form.
So there's like an on switch and an off switch.
They found that hormones like adrenaline and insulin play a crucial role in regulating which form is dominant.
So the hormones are controlling the switch.
Essentially, yes.
Yeah.
Effectively controlling the storage and release of glucose.
This deep dive is showcasing the elegance and complexity of metabolic pathways.
It's a very intricate system.
Yeah.
But it works beautifully.
It's fascinating how our bodies have evolved to such sophisticated mechanisms.
To manage our energy balance. Ensuring a steady supply of fuel for all our cells.
And the chorus's work paved the way for a deeper understanding of metabolic disorders.
It did, yeah.
Like diabetes.
Where that delicate balance is disrupted.
Right.
It's a great example of how basic research can lead to important medical advances.
It's remarkable how all these discoveries build upon each other.
That's the beauty of science.
Revealing not just the individual pieces, but the intricate web of interactions that keep our bodies functioning.
It's all interconnected.
It is. And speaking of interconnectedness, our next stop takes us back to the early days of a technology that has truly transformed how we communicate and connect with the world.
Okay.
Wireless telegraphy.
Wireless telegraphy.
Wireless telegraphy. That sounds like the precursor to modern radio.
It is, yeah.
I can't imagine a world without instant communication. It's hard to imagine, isn't it?
It is.
But back in the early 1900s, the idea of transmitting signals through the air without wires was revolutionary.
It was a huge leap forward.
It was.
Yeah.
And we have a Nobel lecture from one of the pioneers of this technology.
Okay.
Carl Ferdinand Braun.
Carl Ferdinand Braun.
Wow. So we're going back to the very birth of radio communication.
That's amazing. What were some of Braun's key contributions to this field?
Well, one of his key contributions was developing a much more efficient and reliable transmitter for wireless telegraphy.
So he was basically improving the way those early radio signals were generated and transmitted.
He figured out how to use a coupled circuit design to generate those high-frequency electrical oscillations that carry the signal.
What about receiving those signals?
Okay.
I imagine that was a challenge as well.
You're right. And Braun also played a key role there.
Okay.
He developed something called a crystal detector.
A crystal detector?
Yeah.
Okay.
Which use the unique properties of certain minerals, like galena, to convert those high-frequency radio waves into audible signals.
So his work helped to make those early radio receivers more sensitive.
It did,
and capable of picking up those faint signals from afar.
Exactly. And his work on crystal detectors laid the foundation for even more sophisticated radio receivers, eventually making it possible to transmit and receive not just simple dots and dashes, but also voice and music.
It's amazing to think how far we've come.
It is.
Since those early days of crackling radio signals.
Yeah.
This deep dive has really brought to life the stories of scientific ingenuity and perseverance throughout history.
It's been a fascinating journey.
It's inspiring to see how those early discoveries have paved the way for the technology we take for granted today.
It really shows how science builds upon itself over time.
Well, we've reached the end of our deep dive.
We've come to the end.
What a journey it's been. We've explored a mind-boggling array of scientific discoveries.
We have.
From the microscopic world of ion channels to the vast expanse of the cosmos.
It's been a whirlwind tour.
What stands out to you as the most significant takeaway from this exploration?
Well, for me, it's the realization that science is not a collection of isolated facts, but a dynamic and interconnected web of knowledge.
It's a constantly evolving story.
It is.
Yeah.
Each discovery builds upon others.
Yeah.
Sometimes in unexpected ways.
Yeah, absolutely.
Leading to new insights and innovations.
I completely agree. And it reinforces the idea that progress in science often comes from challenging assumptions.
Absolutely.
Asking those what-if questions. And being open to new perspectives.
Even when they seem radical or unconventional.
Right, like those scientists we were talking about.
We saw that with Barry Marshall, who challenged the long-held belief about the causes of stomach ulcers. And with Sidney Altman, who overturned the dogma that only proteins could act as enzymes.
Those stories are a powerful reminder that the pursuit of knowledge requires not just brilliance, but also courage and a willingness to go against the grain.
To think differently.
Yeah, absolutely.
To challenge the status quo.
Now, before we wrap up completely, you mentioned there were a couple of intriguing details from the sources.
A couple of little tidbits
that we didn't have time to fully unpack.
We didn't.